Okay, so in this one, I'm going to be going over the 2016 AP Biology FRQ number one. And this FRQ caused a lot of issues. I remember when it was given this year. And if I'm being honest, I do feel like that this would be something that I think you would see on this year's exam because it's incredibly analytical. There's nothing that you could really look up. So I think this one is worth everybody's time to at least make sure we feel comfortable with how this problem is worded, what you're doing, and let's try to make the most sense of it. And that's actually not too bad. So the passage says leucine aminopeptidase, abbreviated as LAPS, are found in all living organisms and have been associated with the response of marine mussel to change in salinity. So LAPS are enzymes that remove the N-terminal amino acids from proteins and release the free amino acids into the cytosol. To investigate the evolution of LAPS in wild population, the researchers sampled mussels from several different locations among part of the northeast coast of the United States, as shown in Figure 1. The researchers then determined about the percent of the individuals possessing a particular LAP allele, abbreviated LAP94. So basically, they're looking at a specific gene. That gene is going to be determined if they have the enzymes or not. So let's take a quick look here before we start to read the questions and just make sure you guys feel okay on understanding what this is telling you. So basically, we're talking about LAPS, and as I mentioned earlier, they're going to be helping breaking down a larger protein into the individual amino acids. And there's an allele, that allele, the LAP94, is the allele that's going to help make the LAP enzyme. So they sampled at a couple of different sites here. So let's kind of take a look here. And it looks like salinity, the amount of salt in the water, is increasing. So at sample site 1, the LAP94 percentage is only around 13% of that population. But as they started to increase, when they get to like site 7, the percentage of those mussels that are surviving, it's about 59% of the population of those mussels actually have that. So it's pretty evident right here that when you look through this, as salinity increases, the percentage of those mussels that actually have the DNA to make those LAP enzymes starts to increase. So that makes sense. So let's look and see what we had to actually answer here. So for A, this was pretty simple. I just went ahead and made it because you guys are going to have to make a graph on this year's exam. But one thing that you are doing, let me just move this real quick, is you're going to basically construct an appropriate labeled bar graph to illustrate the observed frequency of the LAP94 in the study of population. So basically, it was a bar graph. And again, because you're not taking data over a set amount of time, you had different sites. So site 1, and the only thing that I would make sure you guys feel comfortable with is understanding your dependent variable was the percentage of that LAP94, because that's what they were measuring. So your x-axis was the site, and then your y-axis was going to be the percentage of the LAP94 gene. So site 1 was right around 13%, and then when you get to sites 6, 7, and 8, they're all at about 59%. So that's how that one looked. Okay, so let's take a look at B. Part B says, describe the most likely effect of salinity on the frequency of the LAP94 allele in marine populations. Predict the likely LAP94 at a sampling site between site 1 and site 2. So basically, I kind of already explained this here, as that when you look at the increasing salinity, what happens to the percentage of the LAP94 allele? It looks like it increases. So for the described portion, I would say that if you increase the salinity, you're going to basically increase the percentage of the LAP94 allele, right? So that was an easy point. This next one is kind of interesting, where it says predict the likely LAP94 allele frequency at a sampling site between 1 and 2. Well, when you go here, you get sites 1 and 2, pretty long distance. They want to figure out, well, what percentage do you think those would have here? Okay, now there's a couple of ways that you could have done this. There's this big range that the AP curriculum gave you. If you added these up and divided by 2, that was one way of doing it. So if you did 16 plus 13, you divided that by 2. basically you got a response. You could say for the predict, if I actually added this up, 16 plus 13 divided by 2, that would give me a percentage of 14.5%. That's totally okay, but I want to say that they give you a range of between 14 and 15. So what that means is if you gave 14 to 15%, it any of those would have been correct, as long as it wasn't above 16, as long as it wasn't below 13. That's what they were looking for. So pretty easy couple of points there. So the graph is actually worth 3 points, labeling your axis correctly and then graphing it correctly. And then this is worth 2 points. So you're already halfway there in this one. Now part C. Part C is by far in my opinion, the most challenging part of this entire FRQ. And for part C, it says describe the most likely effect of the Lab 94 activity on the osmolarity of the cytosol. So basically, when you read that word osmolarity, it's its ability to essentially control osmosis, how much water goes in and how much water goes out. Describe the function of the Lab 94 in maintaining water balance in the mussel cell living in the Atlantic Ocean. So let me just kind of jump up here and explain this. Now, it's actually, in a weird way, in a very sneaky way, a water potential question. So let me kind of just revisit what water potential, as well as kind of what osmosis is. So I'm just going to talk about the solute potential because there's no pressure going to be applied here. So it's negative I CRT, right? Where I is going to be the molarity. Excuse me, C is the molarity. Okay. And then I is going to be your ionization constant. So ionization constant in this one is actually 2 because it's salt. But basically, the key thing that you have to know is that regardless, is that water goes to where there is more solutes. So that's critical. Now, let's take a look here at this picture. This is my rendition of a very simple explanation of what's happening in this whole idea of this FRQ, where LAP 94, basically, let's understand what's going on here. So let's say that these 3 that I have drawn here, these are some proteins, and it tells you here, you really had to go back and read this passage to make sense of it. So it tells you LAPs are enzymes that remove the N-terminal amino acids from proteins and release the free amino acids into the cytosol. So that was important for you guys to understand this. So what I have here is showing you that, okay, here is a really saline environment, in the sense that if this was in one of those really salty environments, what would happen here without that LAP enzyme, if there is more salt on the outside of those muscle cells, there's more solutes here, right? I only have 3 solutes here because I have 3 proteins, okay? Each one of these represents an amino acid, and then I have my amino end here. If I have more solutes on the outside, water is typically going to want to rush out because it's in a really saline environment. So the muscle cell would actually start to shrivel up. However, when you actually do the digestion, or actually using those LAP enzymes where you break down all of these proteins into their individual amino acids, and there's always going to be water on the exterior too. So when you break it down, look at what you've done is that you've actually created all these additional solutes. So that actually causes water to want to be rushed in, because normally without the LAP enzyme, if it's in a saline environment, it would actually lose water. But when you break them down into their individual amino acids, that actually helps water to go back in to the muscle cells so they don't die. So describe for C, describe the most likely effect of osmolarity of the cytosol. Basically, the points are going to be given by explaining what I just did, to where if the proteins are broken down, into the individual amino acids, you're going to help water move in. And that's something that's important because without that, the water would leave and these cells would die. So describe the function of lab 94, maintaining water balance in the mussels living in the Atlantic Ocean. So basically what that's telling you is essentially the same thing. Describe that function in maintaining that water balance in cells. You got to understand too, really what you would be looking at here is that the more hypertonic the solution would be in, meaning the more saltier the environment that these mussels are in, the more likely you would get the expression of those enzymes. So let's just go back and double check the rubric on this one. I'm going to go and keep that. And with this, it shouldn't be too terrible. Describe the effect. Lab 94 increases the solute concentration, in the cytosol. And like I said, the function, it just prevents water loss to the environment. So that's what you were putting out, is that what's the point of this? Proteins broken into the individual amino acids, you're increasing the solute concentration. Why does that matter? Where you're causing water to actually rush in. So that is very, very analytical. A really good question on that one. And part D, if I'm being honest, I don't love part D because it's kind of repetitive in terms of what they're wanting you to explain. So it says the marine mussels larvae are evenly dispersed throughout the study area by water movement. As larvae mature, they attach to the rocks in the water. Explain the differences in LAP94 allele frequency among adult mussel populations at the sample site, despite the dispersal of larvae throughout the entire study area. So basically what that means is all across here, they actually added in larvae, which will eventually grow into mature mussels, okay? And as the water moves, these larvae are gonna attach to the rocks. So we have to explain the differences in the allele frequency among adult mussel populations. Now, this one is kind of interesting because as what you had to infer here is that the researchers don't know if those larvae had the LAP94 gene, okay, for the first one. So explain the differences of what you would see if they just randomly put larvae out. Well, basically you had to explain that the mussels with that LAP94 are going to survive in high salinity environments. Okay, so the ones that actually do survive in these salty environments are going to be the ones that have the allele, and then mussels without the LAP94 allele are less likely to survive. So you would get ones with low of LAP94 surviving in like sites 1, 2, and 3, basically what they already received. So don't love it, but that's a pretty standard point to get had. Now you couldn't have gotten 2 points. Hopefully you understand that you can only get one or the other. Now moving on to the predict the likely effect of the distribution of mussels in Long Island if the LAP94 was found on all of the mussels in populations. Justify your prediction. So basically now if they were actually to do this experiment again, but 100% had the LAP94, well, you would see a much higher population growth in sites 6, 7, and 8. And again, the justification, the mussel population will increase in the high salinity. And the other explanation is that they have that gene and they can actually osmo regulate. So they can control how much water comes in and out. Now you could have worded that the reverse way in the sense that you could have said mussel population will decline in low salinity because they have that gene. And they're not necessarily mussels with low salinity will not osmoregulate. So I would have stuck with the first one where mussel population increases in high salinity because they have that LAP94. So those would be the 2 easiest ones. So really good FRQ, especially part C. A and B are really just looking at data, but overall good FRQ.